Across the globe, our environmental waters are impacted by per and polyfluoroalkyl substances, or PFAS. Since the 1950s, these synthetic chemicals have been widely used in a range of everyday consumer products and industrial applications due to their water and grease-resistant properties. PFAS are highly persistent in the environment, taking an incredibly long time to break down, especially in water. Due to their properties, persistence, and widespread use, PFAS are frequently found in the environment where, if not addressed, they have potential to impact humans and wildlife. Reliable, fast, and effective PFAS monitoring is critical to assessing human and environmental exposure to these chemicals and mitigating risk. The microporous polyethylene tube, or MPT passive sampler, has been developed by the Queensland Alliance for Environmental Health Sciences as part of a US Department of Defence funded SERDP project, ER20-1156. In collaboration with research partners, our sampler provides a cost-effective, quantitative technique for reliable and sensitive PFAS monitoring in our waters. Compact, durable, and easy to deploy, the MPT passive sampler measures dissolved PFAS concentrations in surface, ground, storm, and wastewaters. The sampler is time integrative, meaning it enables readings of average PFAS water concentrations over time, rather than just a point-in-time snapshot that is less representative. The MPT sampler is 4 centimetres in length and is made from microporous polyethylene filled with an anion exchange sorbent inside. PFAS diffuse through the polyethylene tube and effectively sorb onto the sorbent phase inside, collecting them for analysis. This versatile device can determine PFAS concentrations, assess spatial temporal trends and environmental fate at impacted sites, and aid in risk assessments. As the MPT sampler is time integrative, it provides a unique understanding of exposures and ongoing accumulation of PFAS in dynamic water systems, offering a more accurate picture of the environmental impact. Our samplers have already been used by government departments and consultancies to assist with PFAS site investigations, leading to better informed decisions and improved environmental management. These collaborations have helped us refine the use of the MPT sampler, simplify deployment procedures, and improve its accuracy and performance across different aquatic systems. As we work to address this global challenge, we're committed to making our research accessible. Our findings and methodologies, including MPT passive sampler deployment guides, standard operating procedures, and good practice guidelines, have been published and are available to all.
Using the microporous polyethylene tube, MPT, passive sampler in the field to monitor PFAS concentrations is easy and simple, allowing you to focus on interpreting the data. Our deployment guides for surface and groundwater sources list the equipment required as well as providing detailed deployment and retrieval instructions. After preparation in the lab, MPTs are stored in ultra-pure water for transport. Before you head out into the field, make sure you've collected all the required equipment. You will need an adequate length of rope or chain and a weight to prevent the device from floating. We recommend testing the rope or chain length and deployment position before getting too far into the process. At the sampling location, secure the deployment rope or chain to the permanent structure or buoy above the sampling site. Gently remove the MPT passive samplers from their plastic containers and discard the purified water. Place the plastic containers back in the deployment container. Then, attach the samplers to the deployment rope or chain using cable ties, checking they are firmly secured. Once the samplers are attached, carefully lower the complete set of samplers into the water, aiming for the mid-depth of the water column. This also applies in groundwater wells, where the slim design of the MPT can even fit within narrow bore wells. Make sure to record all sampling information on a chain of custody form, including sampler ID, location, date, and time. If field blanks are being used, these can be placed beside you while the sampler is being deployed and then returned to the lab after deployment. Leave the MPTs at the sampling site for the duration of sampling, typically 1 to 6 weeks. To achieve greater concentration of chemicals over time, the samplers may be deployed for even longer. To retrieve the samplers, remove the deployment rope or chain from the water, shaking to remove any excess water or adhering suspended solids. Cut the passive samplers off the rope or chain and place each cage back into its respective labeled containers. Store the retrieved samplers in a cooler bag with ice bricks, record the retrieval date on your COC. MPT samplers can then be taken or shipped to the lab for extraction and analysis, or stored in a freezer until analysis. Please refer to our extraction standard operating procedures for more details on extraction and analysis. For more information on deployment procedures or good practice guidelines, visit the website.